In today's episode, we are hosting a wonderful Venom, Professor Tariq Daouda, a specialist in artificial intelligence related to medical sciences, and his brilliant PhD student, Anas Badraoui. Professor Tariq, thank you for accepting our invitation and you're welcome.
Thank you. Thank you, Ibrahim.
Thank you so much. Thank you for you, Anas, and you're welcome in our PhD podcast.
My pleasure.
Thank you so much. So, Professor Tariq, would you please speak about your background?
I had my baccalaureate in Morocco, and this is what and then I moved to France for a few years. I did a licence in mathematics and computer science. And it was a very interesting, very important part of, I would say, of my curriculum, because it gave me a very strong foundation in mathematics and computer science. And then I moved to Montreal, where I did a master's in AI, deep learning, and it was applied to music. So very interesting subject. So I made deep recurrent neural networks that can generate drum partitions.
Okay.
So you give it the style, you give it the beginning, and then you get the whole partition.
Artificial intelligence, we can apply it to anything of any topic. So,
exactly.
Yeah.
It's like mathematics. It's like electricity. You can apply it to anything.
Yeah.
After that, I PhD. I moved to a PhD. So I met my Sébastien, who became my PhD co-supervisor by chance, and we started talking and. He had this idea, basically, that he was looking at that point. It was like AI didn't really penetrate the biology world, but he could see it coming. He's a bioinformatician, and he could see it coming. And we just met basically by chance. And he asked me to come in his office. We started talking about AI and biology, and then I told him that I did AI applied to music. And he was like, okay, if this guy can do AI applied to music, maybe he can also do AI applied to biology.
Absolutely.
So this is basically like the beginning of my PhD. So I applied, I got accepted. So I started a very long PhD, 7 years. The reason why it was very long is because I come from a computer science background, so I had to learn all the biology. So I can be a biologist now and talk to biologists and they understand what I say. That took me 7 years.
Yeah.
And this is where I started working on AI applied mostly to fundamental biology. So using AI to answer basically the deepest questions in biology. So this is what I started doing. I worked mostly on the immune system, what we call the adaptive immune system, which is the part of the immune system that learns to identify viruses, cells that have been infected by viruses, cells that have become cancerous. So that part of the immune system, I applied AI to it.
Good.
And I also learned about the different modalities like mass spec, FACS, all the stuff that biologists use. And then I moved to Boston, where I was affiliated to Harvard Medical School, the Broad Institute, and the Mass General. And there I did still AI, because that's all I do now. AI applied to single-cell sequencing. So single-cell sequencing is a revolutionary technology when it comes to biology, because I mean, it's not that new anymore, but it's still revolutionary. It allows us to sequence cells one by one. Before that, we had to take a whole tissue and then average that, and then you get the average, basically. Take all the cells, you blend them together.
Which kind of cells?
Any cells.
Any cells.
Just like take the cells, you blend them together. After you blend the cells, you sequence that, so you have basically an average of all the cells you have. Now what we can do with single-cell sequencing is that we can sequence cells one by one.
Okay.
And that's extremely interesting, especially for disciplines like immunology. So most of what I did is in immunology, and in immunology you have so many different cell types. These immune cells doing a lot of things in your body, protecting you from all different pathogens, from cancer, for everything bad that can happen to you.
Okay.
There's an enormous amount of different cells, of different immune cells. And when you average them all together, you lose the specificity of the cells. So it's very hard to be able to tell, okay, this cell is doing this thing, this cell is doing that thing. It makes it much more hard to see the whole diversity of the immune cells that you have circulating in your body. So now with single-cell sequencing, we can get them one by one, and we can say, okay, this cell looks very similar to this one. They're the same cell type, but they're not in the same state. So it means that they're probably doing something different. Then we can look at what different thing they do, and then we can from that we can derive, for example, why you have some autoimmune disease, or why this patient survived from cancer and not this one.
Yeah.
And so we can answer these very deep questions about biology.
That's very interesting. I'm gonna come back to you, Tariq, okay?
Yeah.
Now, Anas, snake venom. What's a rare and unique topic? What's actually lead you to follow a thesis in this topic?
Well, that's a very interesting question. I can say it was a coincidence.
Okay.
But let me tell you about my background that led me to snake venom. So the idea, the thesis, come from my supervisor. But before that, I was working on, my background was
All
mainly
come from
mechanical. Yes, it was mainly mechanical design, my background, mechatronics. So I was creating affordable artificial limbs for people with amputation. And they had an experience volunteering abroad in a low middle income country, Egypt,
Okay.
for 45 days, doing, building artificial limbs, both lower extremity and the upper extremity. And that project impacted society because I had the experience to work with 71 person from different ages. And keeping that in mind, I came to my country. I was back then, I'm second year master's degree. And I was impressed by that project, by the contribution it have on society. So I did my final year on that. I created an affordable bionic arm controlled by muscles, not just mechanically controlled.
Yeah.
It was a very interesting project, and I published 2 papers, and I wanted to follow my master's degree,
that's good. Yeah.
to follow my passion. So my idea was, I did a lot of research in Morocco, research about institutions that do that, but I didn't find. I found UM6P, and before the interview, before I applied, I didn't find that subject that I was working on, but I found this supervision.
Okay.
And that's a very important point in your
Yeah.
PhD.
Yeah.
So I did a lot of research about the interviewees, mainly Tariq.
That's actually a critical point we're gonna talk about later.
Yes,
Okay.
yeah, it's a very interesting
Yeah.
point.
Yeah.
So I did a lot of research about my supervisor, and
That's
I was
good.
like, this is the guy that I want to work with.
Good.
So we had the interview, everything went well, and that's how he proposed the snake venom topic. And after that, I didn't know anything about it. I did a lot of research and I found that it has a high impact. So let me tell you about the numbers. Statistically speaking, based on the last survey from the World Health Organization, 2.7 million people are affected by snake bites, and 137,000 people die from it. 300,000 permanent disability, and recently it has been estimated that one person die every 4 to 6 minutes.
Wow,
And who's those people who die? Mainly children and farmers, agricultural workers.
yeah.
So it's a project that have a very high impact, and still people die till now because so far we didn't find a universal antivenom.
Yes.
Oh, that's
There's
good.
also
Yeah.
one interesting thing is
Yeah.
that, let's link the two, is that envenomation also cause a lot of amputations.
Yes.
Yeah.
Yes.
Exactly.
Sometimes the only way to save the person is to cut the limb off.
Yeah.
Yeah.
If we take a look at the venom composition, because venom composition is actually a cocktail, a complex mixture of bioactive molecules. And there are some proteins there that actually lead to necrosis. They do cytotoxicity, they kill cell. And if not treated the right way, you get your amputation if you want to leave.
Yeah.
So that's very critical and alarming.
And how would you actually resolve this problem? How do you do you identify the venom snake or?
I mean, you can't resolve the whole problem by your own,
Yeah.
but you contribute to the research, the things that are going on right now. Actually, we are working on the most critical aspect on venomous species in general, neurotoxicity, that lead to paralysis and in most of the time, death. So, okay, so actually, if we take like a protein from snake venom, let's talk about snake venom, but they are all common. Scorpion, cone snails, they all disrupt the nervous system. So they have points in common.
Yeah.
Some hidden patterns that if you, I can say, if you talk to a biologist, he's going to tell you that if you want to fully understand a venom composition or a single molecule, it takes years to understand, to fully understand the mechanism of action, how that molecule works, the binding effect it does to the neuroreceptors in the brain. So it's very complicated.
Yeah.
So digging deeper into each molecule, we found that there are points in common because they all do the same effect. They all disrupt the nervous system. They lead to paralysis, and we call that the hallmarks of neurotoxicity. Before, I can say it wasn't doable, but now using artificial intelligence, we can make that happen.
Yeah, that's good. Actually, understanding how it works, this protein, that's help in crafting the right solution and.
Definitely, you need to dig deeper into the venom composition, and that's
Yeah.
what we did.
Yes.
Last year, we had with our collaborators, we got a data sets of 130 snake species from different families globally, and we analyzed their venom, their venom compositions, the proteins, families, and stuff, and we found that there are many similarities. And also, we analyzed a specific, very, very important metric. They called it the LD50. It's the little dose that killed 50% of the population. And we found that though the most lethal snakes worldwide that they belong to the Elapidae family, some examples
Yeah.
are the cobra, the mambas, they exist worldwide. They have a low LD50. That's the first thing. But the most important problems that a lot of people should work on right now, they are actually working on, is to finding a universal antivenom. And let me tell you
Yeah.
why. Simply because the venom composition of the same species, let me give you an instance, like a cobra
Yeah.
from Morocco that exists in Sahara and Marrakesh, and the same one from Taiwan, for example. They are the same, from the same family, same species. When you dig deeper into the venom composition, it's completely different. So if you want to create an antivenom that can neutralize that venom, if you create it in another country with that venom composition, it will not work on our local
It's actually
species.
a kind of geographic specificity to each.
Exactly,
yeah.
because there are many factors that
Yeah.
influence the venom
Yeah,
composition, including
Mm.
diet, the weather, age, and the location, of course. It's very important.
I think something is very interesting about Anas
Mhm.
is that when you look at his background, there's nothing in his background pre-PhD that really prepared him for the work he's doing now.
Yeah.
Like if you look at the curriculum, it's not a background in computer science,
Yeah.
and it is not a background in biology.
Yeah.
And I think this is a very important point. Because whatever you did during your master's is minimal compared to the skills you need to acquire for a PhD.
Yeah,
Yeah.
I agree.
It's very small. It's very, very small. So the background you have acquired in these 2 years is not that important. What is more important is how smart you are, how motivated you are.
Yeah. What did you see in him that led you to believe in him?
It's easy. It's easy. It's easy. Look, when you have a young guy in front of you, someone he comes, you have this young person, and he gives a talk, and during the talk he shows you a bionic arm that he made himself.
Yeah.
Like he printed the 3D, he made it, he assembled it, he sourced the material. There's no way you can say this guy is not motivated and not smart.
Yeah.
There's absolutely no way you can say that. This means that this person is probably very good PhD material because it's not something that's easy to do. And this is way more important than the classes you've taken during your master's. Way more important. Way more important. And also, it shows you that because it's a complex problem that you had to solve. It's not easy to make like a bionic arm. It's a difficult project. And difficult projects prepare you to failure. Because nobody can take a difficult project from start to finish and not fail during the process and find
Absolutely,
solutions
yeah.
and keep going. And this is a microcosm of the PhD. Because during a PhD you'll have a lot of failures. Everybody has. Never works.
It's part of the.
It's part of the
journey.
exactly.
Yeah.
And you need the mental fortitude to go through it.
Absolutely. Yeah.
So, seeing that project, I was like, okay, he's smart, he's motivated, he has creativity. He's a creative person, and he can work and fail and keep working until he gets what he wants. That's, to me, this is a good representation of a good potential PhD student. And to me, it is way more important than the grades. Way more important.
Yeah.
So I want to touch upon this subject. Now, if we talk about AI.
Okay.
Okay. So the way we use AI is like a microscope. And this is where I think let's say the revelation hasn't come yet, but it's beginning. It's beginning. AI is not something to replace the biologist, just like the microscope is not something to replace your eyes.
Yeah.
It's something that allows you to see things using your eyes that you couldn't see before. It's an enhancer.
Yeah.
And this is what AI is. Like, no, I don't know any human being who can analyze 200,000 sequences. 200,000 protein sequences or like DNA sequences. That is out of what human beings are usually capable of doing. But if you use AI, it's not AI that's doing the job. You build the AI that enhances your thinking, that enhances your brain, that's capable of extracting what is important from the sequences based on what you think is important. Exactly like a microscope. Because you wouldn't take a microscope and then focus it randomly. You focus it on what you think is important. And this is what we do with AI. We take this framework and then we apply it precisely to enhance human thinking. And when we do that, AI actually becomes a tremendous accelerator for biological discovery. So this is what we're doing with Anas's project. So the idea of Anas's project is, okay, let's take all these protein neurotoxins.
Yeah.
Because it's weird. You have a cone snail. You know what a cone snail is?
Yeah.
It's a snail that looks like a cone. They live underwater. They eat fish, basically. And toxins, neurotoxins, hemotoxins, they're not there to kill you. They're there to help these animals hunt. So if that snail bites you, you get the neurotoxicity. And the protein can be very different from the protein from a snake. But they act roughly in the same way. So the idea is, can we take all these proteins from different realms? Animals, some animals crawl, some animals swim, they're all different, but they all produce neurotoxins. And let's use AI to identify what makes these proteins neurotoxic. Because if you can do that, we basically understand neurotoxicity from the toxin perspective, and then we can imagine a general antibody that will work on all these toxins. Yeah.
Anas, I can see that in addition to your scientific writing, you are passionate about writing in different topics related to the PhD journey, journey. Yeah. So in one of your articles, you talked about being PhD students in Morocco. Can you tell us about this article and what's insights? Can you remind the insights and advice from this article, please?
Sure. So the article titled "Being a PhD Student in Morocco Today,"
Yeah.
and was published in eLife as a feature article. So in that article, I discussed the challenges that are common among PhD students and PhD holders in the global South countries. So some of the challenges when you are working for months and then you apply for a conference abroad in a high-income country, and then your visa gets rejected. That's a big problem.
That's 100%, that's a lot of visa problems.
I talked about the culture and the language barrier. I've had many different kind of informal interviews with many PhD holders in public universities in Morocco and abroad. The majority of them share the same, I can say, reason why they don't want to do a postdoc. Why? Only because of the language barrier.
So we're gonna talk now about a point you have raised before about choosing the best supervisor. But with you, Tariq, please, in your opinion, what's the how can we choose the best supervisor for our thesis?
Extremely important question.
Yeah.
Extremely important question. So it's even more important than choosing the right PhD student.
Yeah.
Because for a PhD student, your PhD is the most important thing you'll do in your life.
I mean, it's just a way to achieve your long-term
No,
goals.
no, no, no.
It's not.
No, a PhD is different. A real
Your
PhD
opinion.
is very different. When you finish the PhD, you're a whole different person.
Absolutely, yeah.
Completely
Yeah,
different
yeah,
person. It's not something you realize until you've done a PhD.
yeah.
And that changes the whole trajectory of your life. It's not like a master's, because it's much more intense, and it lasts much more longer, and you do stuff that are much more harder.
Yeah.
The supervisor is way more important than the project, because the project will change. It will evolve depending on your research. During your PhD, your project will evolve because you discover new things. It's crucial to really choose the right supervisor for you. And one of the best ways, in my opinion, to do that is, of course, you have to meet with the supervisor face to face. If you can do that, not remote, face to face. And then if you can do it, the best way, in my opinion, is to visit the lab.
Yeah.
Visit the people that are already there doing the PhDs, the postdocs, because they will tell you, by seeing them, you'll know what the culture of the lab is. Because the lab is like a microcosm. It's like you live together for a very long time. You spend a lot of time together. And you'll see what the culture is, you'll see if you get along with these people, and you will see if you will fit in that lab. It doesn't matter. If you think you fit with the supervisor, you don't fit with the lab, it's not a good lab for you.
Mm.
Yeah, I
That's
want to
good.
add something
Can I-
about
yeah,
this.
absolutely.
This is very interesting,
Mm.
and it can apply to different labs with history. But for example, let's talk about a lab that has just started.
Mm.
That's a very important thing. But in my opinion, when applying or before applying, is to look for the supervisor online. Just read his papers.
Yeah.
See with whom he publishes, where he publish, because that's how you know if that supervisor aim for quality. Because that's
Mm.
exactly what matters in research, no quantity. That's the first thing.
Mhm.
The second thing, if you're an achiever, you should aim for a supervisor who's achiever, who's better than you in achieving things, and do you see him as an inspiration, you admire him, and that's exactly how you can detect if that if that supervisor is good for you or not.
Yeah.
I mean, there are many things, but if a if a graduate student can't do that before, I can say, in my advice, he's not able to pursue a PhD during that 4 to 7 years. But if he does that, trust me, this is his opportunity, and no one can take it away from him.
Yeah.
It's a very important topic, because your supervisor, such as my supervisor, you're
Yeah,
gonna
yeah,
spend at least 4 years with him,
Mm. At least, yeah.
at least. So it's a very important.
Yes.
It's very,
can-
as
I
Tariq
can-
mentioned, more important than the subject itself. Subject can
Exactly.
change, you can adapt. It's very flexible, but your supervisor
Mhm.
is very important.
We can also say that mental being and mental health is quite important in the PhD journey. Professor Tariq, how do you handle this thing and pushing your students achieving their academic and PhD tasks?
Everybody, it was the same for me, it's the same for everyone. You have some bad habits, you have some bad ways of thinking, and you have to convey that to the student, because at the end, it's you doing it, you always do it in service of the student's project. And when I say project, I don't mean PhD project. I mean farther than that, I mean life project, where the students want to get. So you have to do it this way. And also you have to be very careful not to inject any type of toxicity in the lab.
Yeah.
Never.
Exactly, yeah.
I'm 100% against any practices like, for example, putting 2 students in competition.
Mm.
That creates a lot of toxicity, and that creates a lot of bad behaviors. Because if you look at it, I like to think mathematically also. If you take a system and you say, okay, you 2 people, let's get into game theory a little bit. You're going to get into competition. There's two ways basically you can get better. One, you work harder than the other person. This is what everybody thinks everybody's going to do.
Yeah.
Or secondly, you sabotage the other person.
Mm.
It's way less costly, way less energy expenditure in sabotaging someone's work than working harder.
Yeah.
So you end up in a situation everybody's sabotaging everybody. So never do that. Every student needs to have their own project, based on their own interest, and then it opens up a whole new alleys of collaboration.
Yeah.
Because every student is different, which is very interesting. Because they're different, they will develop skills differently. At the end, they'll all be good at everything, but they don't grow the same skills at the same time, which is very interesting. So someone get better at presenting, the other person will get better at writing, the other person will get better at programming. At the end, they will all be good at writing, programming, everything. But because they tend to follow their interest, it doesn't grow at the same rate. So it means students can help each other.
Yeah.
The student who knows how to program better can help those students program, and then the other ones can show them how to present. And then instead of having a lab with everybody struggling to basically acquire all the skills, basically create an environment where everybody learns from each other.
Yeah.
It creates a much better environment. There's no need for toxicity. Everybody will
spirit.
learn everybody's paper as long as you work on that paper.
Yeah.
It's easy. And also in terms of supervision, it means you have to do much less supervision as a supervisor because they're working together. You don't have to spend 4 hours that person and spend another 4 hours with the other person because...
But you do it in the beginning.
Yeah, at the beginning, you have to do it. But also, I was starting my lab. So usually when you start your lab, you have 4 people in front of you, you have to give them the right skills. But when you already have people in the lab, you have a new person, the other can pitch in also.
Come, yeah.
Yeah. So you start your lab, there's a lot of work. But at the end, if I spend some time with Anas, I know that knowledge isn't going to stay with Anas.
Yeah.
He's going to give it to the others too. And it's the same for the other ones. So it means that as a supervisor, your work becomes easier.
Yeah.
And also everybody's happier.
If you don't pay attention to this, you can kill the lab.
Exactly.
And that's exactly why you see many, like the majority of PhD, like academia in general, globally, everyone is suffering from this exactly. And today, I checked the news and found that there is a publication published yesterday in the Journal of Proceedings of National Academy of Sciences, PNAS.
Yeah.
And it was published today as news in Nature. They say that more than 40% of postdoc researchers leave academia. That's a very important.
They switch to industry.
Yeah. Academia is the smartest people I've ever met. I met them in academia.
Yeah.
But unfortunately, they leave academia for such things.
And as I see, in addition to your PhD task, you are like engaging in writing and extra PhD things. How do you handle your academic task and your extra academic engagement?
Okay. This is a very interesting question, and I'm not going to tell you I'm a multitasker or something like that.
Yeah.
But I'm going to tell you that once you follow your passion, you don't feel that pressure anymore. And also when you put long-term goals, you start seeing that pressure not as a pressure, but as something that is going to change you and make you become stronger and gonna help in your growth.
Good.
So I embrace it. I embrace that adversity, pressure, and because otherwise if I put, like, for example, a small goal of 5 days, what I'm gonna do with these 5 days, maybe I'm gonna be having a bad day. And I wouldn't be able to make it a good one.
Yeah.
Because it's very limited. But if you have a long-term goal, then if you have a bad day, you're gonna make it good.
Yeah.
Because it's just a day.
Yeah.
A day in 365 or 66 days. Dot 5 years. So it's a very important one, and that's something that PhD students at UM6P should do. They should be inspired of the vision of UM6P. The vision of UM6P is always at least 5 years.
Yeah.
It's not 1 week, 1 month, 6 months. No, and you should do that in your life. It's very helpful.
Good. This is actually a question for you too, Professor Tariq. How do you handle, your task?
When you have to deal with something that has a lot of chaos, because research is chaotic. That's in the nature of the thing. It's chaotic, which means it's surprising you all the time. You have an experiment, doesn't go the way you think. Or you find something new that's way more interesting than what you were thinking about.
Yes.
So it's always surprise, surprise, surprise. So you cannot planify research. At least you cannot do it, you cannot micromanage research. It's impossible. There's no way you can do that. You can micromanage engineering, but not research. It's completely different. So it means that you have to free your time as much as possible so you are able to handle this chaos when it comes. So it means if you, instead of having like specific time that you allocate, it's better to free as much time as possible. For example, free the time that you spend taking notes. Remove that. The time that you spend reading notes, remove that.
Yeah.
That's a lot of time, because if you add that all together, it's a lot of time, especially when you have 5 projects plus the rest. It's a lot of notes. And the thing is that when you write something, also you tend to forget it.
Yeah.
I think it's the opposite. People say when you write, you tend to remember. I don't think so. When you write, you tend to trust the paper, and then you forget it. So that's one of the things. The other thing is almost 0 meetings. The minimum meetings, number of meetings. And why? It's very simple. Like, for example, let's use an economic perspective. Let's say we simplify. I'm worth 10 dirhams an hour, 10 dirhams an hour, 10 dirhams an hour. Very simple, very simple. If we sit all together for 1 hour, we're paying 30 dirhams. That's what it costs, basically. Obviously, anyway, it's way more than that, but anyway. So 30 dirhams. If you and I are having a meeting, and we are in the meeting and Anas is just sitting there, we lost some work time. And also, it's when you extrapolate that to a whole lab over a whole year, it's a lot of work hours that you lose by doing meetings. So it's very important that when you have a meeting, everybody in the meeting should participate or should benefit in some way. And the discussion always has to be very precise. You don't talk about, I mean, Anas, you see how we do the lab meetings. It's like very specific questions.
An efficient way.
Very efficient. You go this, and that. You don't waste everybody's time, and everybody should participate, and everybody should benefit.
Let me tell him an example. No more than 45 minutes, the lab that we do every single week, and it's concrete. The progress shared between all the lab, and everyone needs to give his added value, a suggestion. So as a PhD student, you read that paper before, you read that draft and stuff, and then you come up with, and that's exactly how you can make, Tariq is making the lab not competitive, not toxic, and very productive.
That's good message we finish with.
Thank you, Ibrahim.
Thank you very much, Professor Tariq, for accepting the invitation and for sharing your insights and advice. Thank you very much, Anas, for sharing your advice and insights as well. And thank you very much for watching us, and stay tuned for our upcoming session with new guests and with new experts for sharing knowledge about the PhD journey.